and I learned a few interesting things about chimps. One of the things is that they're very similar to humans in the sense that they like to be tickled.
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and this is the 170th episode of this podcast dedicated to anything and everything that you can do to improve, prolong, and generally get a lot out of the workings of your own brain. If you're listening to this podcast, odds are pretty good that you're a human, or at least a house pet belonging to a human, which means that you've got, inarguably, pretty much the best brain in the animal kingdom. There's a lot of trash talk that happens about the human species, but pretty much everybody admits that our brains are number one. All that being said, this is going to be an episode about animal brains. A lot of us love animals, myself included, and of course it's really interesting to look at where they differ from us as far as how they perceive the world, how they think, and also where we find commonalities. Whether we're talking about pets or wild animals or cartoon animals or dinosaurs that we're speculating about what they might have been like, animals have a perpetual fascination for a lot of us, and we're finally getting around to an animals episode. We're going to be talking to a researcher who studies animals and exactly how they think. His name is Dr. Aaron Blaisdell. He's down at UCLA. Works primarily with rodents and pigeons, but he's gotten his hands dirty with chimpanzees and other animals as well, and we'll be talking with him in the main interview. If you hang around until the very end of the episode, we're going to tell you some oddball facts about various members of the animal kingdom that have amazing things neurologically going on, a couple of highlight reel standouts among the animal brains, saving that for the end in the ruthless listener retention gimmick. But as usual, let's kick things off first with This Week in Neuroscience, and yes, this will be an animal one too.
Smart Drug Smarts, This Week in Neuroscience.
So oftentimes when you're thinking about animal studies and the types of animals used, especially when you want to compare this up to a human, chimpanzees, or at least something in the great ape family, are considered to be kind of the best you can do as far as approximating humans. And the reasons for this are fairly obvious. We are closely genetically related to chimps as opposed to, say, lab rats or paramecia. But according to recent research published in the journal Animal Behavior, in at least some areas of cognition, it might be that humans have more in common with dogs than we do with chimpanzees. They gave a battery of social cognition tests to 2-year-old humans, dogs, and chimps that basically involved hiding treats and then giving nonverbal cues as to where the treats could be found. The humans who knew where the treats were would do things like point or gesture or just look with their eyes. They had 100 or so 2-year-old humans, 100 or so chimps, and about 500 dogs that participated in this study. Needless to say, these were not all in the same place at the same time. The chimps were on a game reserve in Africa, and the dogs were mostly at the Arizona Canine Cognition Center at the University of Arizona. But it turned out that while the chimps performed admirably on tests involving their physical environment and spatial reasoning, they didn't do nearly as well when it came to tests of cooperative communication, such as figuring out what the nonverbal gestures from the humans who were giving the cues actually meant when it came to finding these treats. Both dogs and children outperformed chimps, and they did so by similar amounts, and researchers also observed similar patterns of variation between individual dogs and individual children. Explained the study's lead author, Evan McLean. What we found is that there's this pattern where dogs who are good at one of these social skills tend to be good at a lot of related social things. And that's the same thing that you find in kids, but you don't find it in chimpanzees. Our working hypothesis is that dogs and humans probably evolved some of these skills as a result of similar evolutionary processes. Probably some of the things that happened in human evolution were very similar to the processes that happened in dog domestication, said McLean. Not to be underestimated is the fact that modern dogs were guided by humans in their evolution, not with some sort of grand design, but certainly based on selective breeding for the dogs that would be most conducive to living around humans and being useful to us. This meant that being receptive to human nonverbal cues was probably a defining factor into what led to dogs being dogs. Despite the apparent similarities in social intelligence, there's a lot of ways in which dogs still don't make a very good model of human cognition. The kind of intelligence that we think is very important to humans is social in nature, and that's the kind of intelligence that dogs have to an incredible extent, said McLean. But there are other aspects to cognition, like the way that we reason about physical problems, where dogs are totally dissimilar to us. So we'd never make the argument that dogs in general are a better model for the human mind. It's really just this special set of social skills.
Smart Drug Smarts, the podcast so smart, we have smart in our title, twice.
Picked up a couple of 5-star reviews on iTunes. Listener IPA Brews said, "Great podcast, very interesting and informative, and I specifically enjoyed the nicotine podcasts." Good call. That was with Dr. Neil Grunberg. I like that one too. And Iron Mike 25 from the USA says, "This is a great podcast that I use to fill in my day amidst my fitness-related life. So important to have a healthy brain that I can use to better myself and my practice. Well, a big thanks to both of you. Thanks to everyone who has left iTunes reviews. And of course, anybody that's left anything nice anywhere. Even left the podcast playing when you were out of the room and your pets were still listening. Every little bit counts. But admittedly, podcast reviews are particularly nice. We have got a newsletter at Smart Drug Smarts. You might notice that we put out a special edition podcast in between the last couple of episodes. Went above and beyond the Call of Duty, putting out audio, but have been slacking on the newsletter the last couple of weeks. I apologize about that. You can send your hate mail to Jesse at smartdrugsmarts.com, or if you want to sign up, what a sales pitch that was, I know. You can do so at smartdrugsmarts.com/newsletter. We've got a couple of supplement stacks that we like to both use and spread the word on around here. Those can be found over at axonlabs.io on the web. They go by the name of Nexus and Mitogen. Nexus is an aniracetam-based stack, stacked with a choline, some phosphatidylserine in there. And Mitogen is a mitochondrial support stack. And yes, those do stack nicely together. People often ask if they buy both, hey, should I take both at once? What's the ratio between one and the other? I always recommend that if somebody gets both, to start on one for a couple of days, try it in the absence of the other, and then vice versa. I'm always a fan of breaking stuff up and trying to isolate what effects you're getting from which inputs. Personally, I don't take Mitogen too late in the day. I just find it more of a physically invigorating feeling. Whereas Nexus is more of an any time of day thing for me, but it depends on what I'm doing during the day. Again, it's more of something I would use for like a whiteboard session, trying to come up with big ideas, creative thinking, not necessarily I know exactly what I need to get done and I just want to sit and hammer the keyboard. All of which taken together sounds like a non-prescription prescription, but kind of try it and see. Both Nexus and Mitogen are over at axonlabs.io, or you can click over from the shop link on the Smart Drug Smarts website.
Smart Drug Smarts.
You know, I know it's a little bit controversial, but I swear to you, I've seen drunk birds eating fermented mulberries in south central Pennsylvania.
Those were the words of Dr. George Koob, who is actually not the interview guest in this episode. He's an interviewee in a different episode that will be coming up soon, but he ventured off into talking about animals there, and it just seemed so appropriate that I wanted to get it in here. Kind of showing how even when you're talking about other things with scientists who study the brain, animals keep creeping back into the conversation. A, because they're interesting and cute and we all kind of love them, but also because animal research is part and parcel of finding out how living things work and at least making some educated guesses on how the human brain and body might work. There's obviously a lot of research that for everything from lifespan to size of cages to ethics, you just can't do on humans that well, and oftentimes animals are a acceptable stand-in. And of course, sometimes you just want to look at animals because you want to look at animals. In this interview, I'm going to be talking with Dr. Aaron Blaisdell from UCLA, a guy who's got one of the cooler website names I've ever heard of. His web address is pigeonrat.psych.ucla.edu, and he runs the Comparative Cognition Laboratory. He's also part of the Comparative Cognition Society, basically looking at what we're going to be talking about in this conversation, which is what are the lines between the types of cognition that are available to various members of the animal kingdom? Where are the fuzzy lines between what sort of thoughts and perceptions some animals might have that others might not have? What might it be like to inhabit the brain of a bird or a bat or a cockroach or an octopus? So this might be a little less prescriptive as far as things that you can do to your own brain following what you hear in this episode, but might lead to some interesting conversations that you can have afterwards with your dog or your cat or your goldfish, or if nothing else, might just make you extra appreciative of your human brain. So with no further ado, let's jump in with Dr. Aaron Blaisdell.
As a kid, I always loved animals. So I've had cats, I've had parakeets, I've even had hermit crabs and all kinds of critters and stuff, garter snakes. And when I went to college, I actually got involved through the anthropology department. I was an anthropology major and working with some primates in a primate lab. And that kind of rekindled my interest in what's going on in the animal mind. So I was kind of an assistant to the trainer in that primate lab. And they had two chimpanzees. They were very young females, about 3 to 4 years old. And we were training them to go through different kind of behavioral tasks, reaching for things, walking, and so they can study the natural movements of these primates. I got a lot of time to go in and just play with the chimps, which was a lot of fun. And I learned a few interesting things about chimps. One of the things is that they're very similar to humans in the sense that they like to be tickled. I learned early on that if they're like sitting on my lap and I reached up on top of their neck, squeeze it as if you're giving somebody a shoulder rub, they would start laughing. They would go, this kind of chimpanzee pant laugh. And they would take their hands and try and pry my hands off of them. You know how like when you're tickling somebody and they try and stop you? And so I would stop tickling them. But then a moment later, they grabbed my hands and put them back onto their neck. They wanted me to do it more. Unlike most animals I've ever interacted with, they were actively seemingly recruiting me to tickle them, to do something to them. So that was one of the things.
It's got that interesting aspect too that you can't effectively tickle yourself. I'm not sure if it like requires theory of mind, but I mean, is definitely a two-being activity.
That's true. And that's a good point to bring in like theory of mind, because that was another thing that we observed is we gave them mirrors. And you're probably familiar with the mark test that Gordon Gallup developed back in the 70s. He was interested in understanding whether chimpanzees did have some kind of sense of awareness, which is kind of a precursor for consciousness. And one way that you can test awareness is by giving a mirror. And if you recognize your reflection as being that of yourself, then you can use it to groom yourself. And what he did was when the chimpanzees in his laboratory underwent general routine health checkup where they had general anesthesia, so they were unconscious, he would take the opportunity to place a colored mark, like with some nail polish or something, but normally it was removable, on their forehead in a specific spot. He would also, as a control, place a clear liquid kind of mark, another spot on the face. And then the chimps would come back out of anesthesia and kind of get back into their normal selves. And then he'd watch them for the first hour after they were completely recovered from the anesthesia. He'd watch them and see how often did they touch the area where he put the markings. Mm-hmm. And it was very low, like maybe a couple times accidentally so during the hour. Then he would give them a mirror and he watched how often they touched that marked spot again. And now the times that they touched the colored mark dramatically increased, whereas the amount of times they touched the non-colored mark didn't change. It was still very, very low.
Yeah.
That was kind of his evidence that they understood that the reflection they were seeing in the mirror was one of their own selves, and that, oh, wait, now there's this mark on my forehead. And instead of reaching up to the mirror and touching it, they reached up to their forehead, using the mirror to guide their pointing, and touched the mark. So that was kind of an observation that led to a whole wave of searches, really, for this kind of passing the mark test, as it's called, this kind of aspect of awareness in other species. And some species do, and most actually don't pass this test. And so we actually had mirrors also, and we gave our chimps mirrors just for enrichment purposes. And darned if they would use them all the time. And you know what they use the most often to do? They would stand kind of in front of it, facing it, and they would open their mouths and they'd be looking around at like what's inside their mouth. And they'd reach in and like pull at their teeth. And, you know, when you're looking in the mirror and you're flossing and you're kind of looking at the condition of your teeth, they were really, really curious about what was inside their mouth, which I thought was really cool.
Most of your studies have been dealing with rats and mice and animals of around that rung of cognition. Can you tell us some about that in your recent work?
Yeah, so that early work with primates was just kind of getting my cognition wet for doing that kind of work as a living, as opposed to just a hobby. So I got my PhD in an experimental psychology and behavioral neuroscience laboratory, and then I was offered the job at UCLA, where I currently am a professor. And in the intervening time, my PhD was on learning to study cognition in the rat. And the way that we do that is we use instrumental and Pavlovian conditioning procedures. Those are kind of the basic associative learning procedures. You know, like the Pavlovian conditioning is when a cue predicts some kind of reward, then the cue will induce a kind of reward-related behavior in the animal. So for a rat, if you have a light that comes on and then food's delivered in a little food hopper, then after a few times pairing the light with the food, whenever the light comes on, they'll run over to the food hopper to retrieve the food. And then instrumental learning is something like if they have a lever and every time the lever is pressed, food is delivered, then they will learn to press the lever to get food.
Right.
So those are real basic procedures that have been studied, for 100 years now. Well, basically the operant type with the lever, since Skinner developed that procedure back in the last century. But what we can do, and what I started doing in the laboratory where I was doing my PhD, which is at SUNY Binghamton, is to use those procedures of behavioral associative learning psychology to get at the mind of the rat. And of course, when I say the mind of the rat, that raises a lot of red flags among some of my associative learning brethren who bristle at that terminology.
Yeah.
But really what we mean is that possibly some kind of representation that the central nervous system, the brain, is doing. And these computations on those representations that allow the organism to deal flexibly with the world around them. The way they encode the world and the way they then act on that information. And so that's kind of what I mean by the mind. And so that's what I study.
And even when we're talking about human minds, there's a big fuzziness around what people's definition might be. We have an intuitive understanding that's pretty easy for common speech, but when you try to peg that down scientifically, there's still a lot of various definitions out there.
Oh, absolutely. So without getting bogged down into all those definitions, we'll just treat it like the way that Supreme Court Justice who said, I don't know how to define pornography, but I know it when I see it.
Yeah.
So, of course, we have to have operational definitions when we're working in the animal. animal, especially the non-human animal, about what we mean by some kind of mental construct or cognition. And we have those. So I'll just talk about one of the experiments I've done recently that is really exciting in getting both into the sophistication of rat cognition as well as something we've learned about how it's related to brain function. So let's say that you learn that there are two cues that often go together. You see a light and you hear a sound. Take thunder and lightning, actually. So we know that lightning occurs and it's often followed by the rumble of the thunder, right? Now, let's say that you're sitting indoors and there's no window in the room that you're sitting in, but you hear thunder rumbling. What do you imagine just happened?
There's probably lightning outside somewhere.
There was probably lightning outside. Now, you didn't see it, but you can make that inference that there was lightning, right?
Yeah.
And you can imagine that if you wanted to know, is it raining because I'm about to have to drive home? Do I need to go get an umbrella? You might go to another room where there's a window so you can look out to see, oh, I see it's raining. Okay, you're right. So I heard the thunder. That meant there was lightning, which meant there's a rain cloud. And yeah, it's raining. So those kinds of inferences, because those are all related, associated events. When you hear one, you kind of imagine the other must be present. And if you can't see it, then you might go and do an action to try and verify it, right?
Right, because it might not be 100% that it's raining at that moment, but the odds have just gone up significantly because you heard the thunder.
Exactly. And so that is the kind of question I wanted to investigate in rats. Can rats also understand that if they hear one event, then they'll expect that another event that in the past has been associated with that first event will be there, even if they can't see it.
Mm-hmm.
So the idea is, can they imagine that the other event is present?
Right.
And so we just basically took our standard rodent conditioning chambers, and what we did was we just trained them first that a light and a tone would come on together, and that if the light and the tone came on together, there would be food.
Okay.
So that's the first thing. Then what we did is we tested them in one of two conditions. In one condition, we didn't change the box at all. We put the rat back in the box where it had been trained, and now we just turned on the tone so they could hear the tone, but the light is not turned on. So here they hear the tone, and they look up, and they could see that the light is not on.
Mm-hmm.
And in this case, the rats do not look for food. So they kind of recognize that, okay, the light that normally predicts food is not there, so maybe there's no food. I'm not going to look for it. Now, that's one test condition. The other test condition, we also put the rat back in the box, but now what we do is we cover the light bulb where the light had been presented during training. We cover that light bulb with an opaque shield. So this is like you now being in the room where you can't see outside. And what we do is we present the tone. And now the idea is if the rat hears the tone and remembers that that tone always occurred in the presence of the light in the past, will they imagine that the light must be on? And then because they imagine the light is on, they also should expect food. And so what we expect is that if that happens, they're going to go and look for food in the food niche at a lot higher rate than the rats that are tested with the tone and no light on.
Yeah, with the light demonstrably off.
Exactly. So it's, in one case, the light is unambiguously off. In the other case, it's ambiguous. And based on their past experience, it's probably on. And that's exactly what we found. When we cover that light and we present just a tone, now they act as if they think the light's actually on. Even though they can't see it, they can't verify it.
Do they seem iffy about it, or is it exactly the same rate of giving it the benefit of the doubt as if it were on?
Right. That's a good question. We've actually done a few different types of procedures to investigate this type of an inference that they make about an absent event, or an event that they can't determine whether it's present or absent. And the example I just gave you is actually one of the strongest cases. They really respond when we cover the light and present the tone. Their expectation of food is almost as strong as if we actually physically presented both the light and the tone.
Yeah.
Whereas if we present the tone and the light's ambiguously, demonstrably off, there's almost no expectation of food. So that was the strongest condition.
Yeah.
We have some other procedures where the response is more kind of in the middle, intermediate, sort of like they're unsure and maybe it's on, maybe it's not. So their response is intermediate between whether it's really on or really off.
It would kind of be surprising if it weren't that way. I'm thinking if you were like a rat, the joint triggers of seeing a cat and smelling a cat. If you smell a cat on the breeze, you better at least hunker down and look around because odds are you're going to see one even if you haven't seen one yet.
Exactly, yeah. And you tend to find that kind of phenomenon in the wild. What's interesting about this is that they make different responses whether they can tell that it's off versus can't tell. This is the first study to really unambiguously, pun intended, tell us, researchers, that they really do discriminate situations where they are certain about what they can see or can't see, the information in the world, versus where information about the world is ambiguous.
Right.
They really do discriminate those situations. And that's the kind of the novel feature of the research we're doing.
It seems like there's a lot of animals that even try to sort of game the system, like a possum playing dead, octopuses changing color and things like that, where animals are trying to fool one another and present bad cues to drive the behaviors of other animals. And if animals weren't smart enough to make some of these imaginative leaps, the world wouldn't be nearly as interesting a place.
That's a really good question. And the species that would come to mind would be something like a bear.
Yeah.
Because I know that's one of the things they tell you on the West Coast and you're hiking. They tell you, if it's a grizzly bear, don't run. You'll never get away. You just play possum. And that's the best chance you'll have, is if it just thinks that you're dead because you're not moving, despite having it, nuzzle you and paw you around. If you can really convince it that you're dead, then they'll leave you alone, from my understanding. That's your best chance. You got to trick the bear.
Yeah.
We just published some work a few months ago. The work took a few years to do, showing that it's actually the hippocampus, an area of the brain involved in spatial reasoning, spatial cognition, as well as memory, especially episodic memory, is critically involved in this process of imagining that the event is on when it's ambiguous, like when it's covered.
Yeah, and I guess that would make sense that it's an episodic memory related task, because episodic memory has often been theorized that we use some of those same neural hardware to imaginative fake memories about what the future might hold given this behavior or that.
In fact, that was one of your previous podcast guests, Lucy Cheek, was talking about that, an alternative view of episodic memories. Maybe it was actually it's a byproduct of a selection for a type of process of counterfactual reasoning, imagining alternative scenarios. And so this is akin, the work we're doing with the rats is akin to that, imagining what are the possible states of the world and how do I behave according to those possible states.
What do we see as far as the types of animals where some of these causal reasoning scenarios start to break down? Like sea sponge, we don't expect much from cognitively. Rats obviously can do a lot more.
That is a great question. And if the funding agencies were up to it, they'd be funding that kind of comparative work, because we really haven't investigated that as a field. The comparative cognition has not really investigated this levels of causal representation and causal inference. I mean, obviously we have a lot of work on the kind of more reflexive and also the instrumental learning to press a lever to get food. So there's a lot of work on that causal relationship that animals seem capable of. And even invertebrates are capable of instrumental learning. So they can learn causal relationships. Oh, doing this action causes that outcome to occur. Mm-hmm. Honeybees can do that. Crabs can do that. Other invertebrates can do that. But the kind of causal reasoning that I've been studying in my lab is a little bit more sophisticated than that. And it goes beyond merely kind of the prior learning, stamping in kind of the behavior.
Yeah.
And it's more of a flexible inference beyond what they've learned and deducing novel relationships. Been enough research really in different species to look at that.
Interesting. Yeah, that does seem like a hole in the studies right now, just because it seems like if we had all the data, like what's the hippocampal volume as far as number of cells in these different species, which ones can do these certain behavior levels, with a large enough data set, you could probably draw some interesting findings out of that.
Absolutely. And I put my money on the cephalopods, especially like octopus and squid and cuttlefish, as probably being capable of similar levels, sophisticated computational levels of causal reasoning as rats, because of the sophistication of their behavior and of their nervous system. They're actually very sophisticated animals.
I was reading about that a little recently, and I know that there's a book, I think it's called The Soul of an Octopus, specifically talking about within each one of their suckers, they have 1 million neurons. Compared to 80 billion in the human brain, it doesn't sound like a big number, but it's pretty significant. They have a very distributed intelligence throughout their body in the way that vertebrate animals really don't.
That's right. And not only is it distributed, but they actually act independently. It's almost like a swarm or a hive kind of interaction. With more of the arms go one way than the other, each arm acting independently, then the majority rules in terms of what the overall and the choice of the direction that the octopus will go.
Wow.
So it's hard to imagine what it would be like to be an octopus, knowing that, is it still kind of the central I as the actor? Is that we kind of have that feeling of being the agent of action? Or is it because of the distributed nature of the control systems of their central nervous system, is it more disembodied? It's a good question.
I would love to ask your thoughts about some of the animals that have larger brains than humans. That's really interesting to me that there are animals with much bigger brains than us and that we still seem to have the cognitive edge on them.
Yeah, somebody who I've gotten the most education about these kinds of questions, these are comparative neuroanatomy questions, is from Professor George Streeter, who's a professor at UC Irvine. And he's written a book on vertebrate brain evolution. And it's that book that really taught me a lot about these kinds of questions and general principles of how brains scale. The elephant or the whale, larger whales especially, have more neurons. Because they have larger brains, they have more neurons than we do. It's not that when the brain evolves to get larger, each neuron, each neural cell also gets larger. The neural cells tend to stay about the same size across all the different sizes of species. So therefore, a creature with a larger brain actually has more neurons. And so the question is, well, if it's just sheer number of neurons, isn't that kind of the driving correlation with intelligence? And it turns out that that's not quite true. Although they have more neurons, it seems like the human brain also has more specializations than the brains of other very large brain species like elephants and cetaceans. And then it seems like our frontal cortex, the part of the brain that really kind of handles the more flexible reasoning, the ability to control our behaviors and plan for the long term and things like that, is much more differentiated. There are many more submodules within ours than there are within other species that have equally large or larger brains than we do in absolute terms. So I think that's part of it.
In human evolution in the past, I think it's like 6 million years since we branched off from the chimp, it is the frontal cortex where we've gotten most of the growth of our brain. I think the brain something like tripled in size, but that hasn't been a scaling up equally across the board. Correct me if I'm wrong, but I think it's mostly growth in the frontal cortex that has been the biggest change there.
That's pretty much correct. And in addition to that, actually, there's also more direct connections between cortex and spinal cord in humans than in chimps, particularly for the parts of the brain that has spinal connections to hand and lip and throat motor areas. Because you think about it, we're much more dexterous with our hands, our opposable thumbs, and our ability to really fine-tune manipulation skills with our hands,
right,
as well as speech. With the kind of motor control you need for speech is dramatically improved in humans compared to chimps. So tool use that started about 2.5 million years ago with the origin of genus Homo, and speech, which is a more recent development, presumably, but still, both of those, there's more direct connections between motor cortex and the spinal innervation of those motor areas. So yeah, we have those adaptations too.
When people are studying brains, we see within humans a lot of specialization between the left and the right halves of the brain. Is there a parallel there within the animal kingdom, or is that something which is more human specific?
Again, it's not an area I've delved into myself, but from my reading, it looks like it's common amongst most species in which it's been studied to find lateralization. In fact, I was just reading a paper the other day where it was comparing dagus, which are a South American rodent, with some other kind of rodent, and I forgot what kind of rodent it was. And it was really weird because for spatial abilities, for one of those species, there was lateralization in function. For the other species, there was not lateralization of function for spatial. But for social interactions, in the other one, there was lateralization, but not in the first. So I forget which one had which, but it was like there was a double dissociation in terms of how the brain, whether or not it was laterally controlling certain behavioral processes, certain domains of cognition, depending on the species. And the authors were trying to tie it to the life history and ecology of the different creatures, which differed. So there's a lot of flexibility. That's one of the amazing things about brains is they're so adaptable and in such interesting and unexpected ways. And I think another thing in the paper, there were sex differences for one of those things, where it was lateral for females, I think, but not for males or something like that. So there's so much variability in how brains adapt to meet the needs of the world for a particular creature.
Back to maybe more of your core areas of study, the history of science over the past 500 years has a lot of ways been stripping away the areas in which humans are special. We no longer think the Earth is the center of the universe. We no longer think that humans were anointed by God in his image. The human mind is maybe the last bastion of human idealism or something where we really kind of think of ourselves as the top of some existential pyramid. In a way, the kind of things you're studying are chipping away at that foundation. Where do you see humans as still having something really distinctive about us, or is this just sort of a blurry-edged topic?
Well, yeah, I mean, I am trying to chip away at that boundary and seeing to what degree do rats and pigeons as well, other non-humans, kind of approximate some of the basic processes that we see in humans, especially at the more higher level sophisticated processes. Nevertheless, what we do know is that humans, there's this one Rubicon we've crossed that seems to separate us from the other animals to a huge degree, and that would be language, and what language provides as a tool, as a mental tool.
I think that's something that as a human, it's so easy to forget about, because learning to talk is something that we do even before we start laying down episodic memories for the most part. But the fact that most animals just eat with their mouths, and that we've got this mouth which has these awesome cross purposes, we can still eat with it, but it's our primary communication tool, is really, really something. It's not surprising that there's a lot of neural hardware devoted to that.
That's right. There's no other creature on Earth that has a symbolic form of communication that they learn during development that is completely divorced from the physical world. So the word dog doesn't have to sound like a dog. It doesn't have to look like a dog.
Right.
When you write it or spell it in a Chinese form or Arabic or English or whatever, they can look completely different, but they can refer to the same external referent object, right?
Yeah.
Or idea. And then we can use those to build up abstract level ideas beyond any level, because they're tied to these concrete tokens that allows us to extrapolate and build on those to create abstract levels of thought way beyond what's possible without it. Without it, you're kind of stuck in the real world with your experiences, basically with the experiences you observe and can piece together and make inferences about. One, you can't transport those to another mind like humans can. So we're building on our cumulative culture and ideas all the time. And two, you don't have a level of representation with the symbolic system that allows you to go way beyond what other animals can do. And I think there's some recent discovery that dolphins actually do have some symbolic reference vocal communication system. So they have some of the elements of symbolic language-like communication. But even there, I doubt that it would have the kind of recursive structure, the Chomskyan type of, you can say anything in a million different ways, or you can almost say anything in the language. You just have to be able to put the symbols together in novel ways. I doubt they have that kind of ratcheting up that we have.
Yeah. Things with dolphins, one of the things that makes me so sad is it seems like the way that we're polluting the oceans, it's like all dolphins are probably functionally retarded compared to a dolphin of 500 years ago before the oceans were as polluted as they are now.
That's happening to every species on Earth, including ourselves. That's actually some of the work I've been doing on diet and cognition. And about 8 years ago, I discovered the low-carb paleo diet, and it did wonders for me, especially my mental functioning. My mental clarity was even throughout the day, just felt much better, clear-headed, full of energy in a way I hadn't most of my life. And now having kids, I see the kind of foods that a lot of their schoolmates have, that the parents give them, and compared to the kind of food that we cook at home. And it irks me to no end. But every time I try and talk about this kind of topic to people understand it, but nobody really in the academic world until recently has been paying a lot of attention to that. It's always been focused on, are people getting enough omega-3 or is something like that? But I think the overall diet quality, really processed foods versus whole foods, and the effect that might have on cognition and seeing all the cognitive problems that might manifest in children, especially, who are more vulnerable to these things. It got me interested in trying to develop an animal model so I can look at causal effect. I can manipulate diet and see, does that causally affect their cognition, as opposed to just associative level data that we have from human epidemiology, for example.
Right.
And so what I did was I took two groups of rats, basic outbred strain of rats that we typically use in our experiments in the lab. I had kept one of them on their healthy control diet, which is actually made up of relatively unprocessed ingredients. I mean, it's processed into a pellet, but it's like ground fish meal, ground flax, ground cornmeal, and it's pretty healthy. And they stay pretty healthy in the laboratory eating that kind of diet.
Yeah.
And we did is took a second group of rats and put them on a highly refined, processed, a purified diet. And so the ingredients all from like really refined, purified sources. So it's kind of like emulating that typical high sweet junk food diet that a lot of humans, especially I see a lot of children, really gravitating towards. And it was comparing that to the whole food diet. So they actually were relatively matched on macronutrient ratios, that is the amount of fat, carbohydrate, and protein that compose the diet, but they differed dramatically in how processed those ingredients were, being more at the whole foods end of the spectrum or incredibly refined and purified end of the spectrum.
Yeah.
And this refined diet, one, it was obesogenic. It caused the rats to gain a a lot of fat mass. Pretty quickly, within 2 months, they were a lot fatter than the rats on the control diet. And what I did was I put them onto different types of cognitive tests to see, well, how is it affecting their cognition? And what I found was that, one, it impairs their motivation, that they're not willing to work as hard to get a reward, whether that reward is like a little bit of sweet sugar water or just plain water.
Yeah.
So they're much more what we might think of in human terms as lazy. So refined food seems to make them lazier, less willing to work hard to get something. And two, I found that they have an impairment in their ability to sustain their attention and to focus on their attention, to wait for a signal to get a reward.
Yeah, both of those sound like dopamine-related changes.
Yeah, they could be. In fact, another lab has actually shown that more Western-style diet, as they call it in the rodent work, which is high in fat but also high in sugar, does affect dopamine receptors in the nucleus accumbens, a part of the brain that's a part of the reward system. But also the attention effects, I think, are probably related to frontal cortex behavior because they were also more impulsive. They were less able to wait for a signal to come on before they make a response, and they were making many more premature responses.
Would you characterize that as being almost like the rat equivalent of ADHD, like in a kid in a classroom?
Yeah, I would say so. And I think that the implication then is that if rats are given this really processed food diet, it's high in sugar, and they're getting ADHD-like symptoms, and we see what kids are eating a lot of that kind of stuff, and we know that ADHD is at an all-time high in terms of amount of diagnoses, then maybe the diet is contributing to that to a great deal.
Even a rat could make that causal inference.
Nice. I like it.
What would you like to see as far as follow-up studies to either further refine that point or hammer it down or knock any holes in it?
Well, I think that the things I'd like to see, if I had funding to do it, would be a couple of things. I'm not as interested in looking at all the different brain mechanisms that are affected because we know that there will be brain mechanisms affected. And what I'm actually more interested in is knowing what's the extent of what's affected. Are there any kind of cognitive tasks or learning and memory tasks that are not affected? Is there anything that's spared by eating a junk food diet? That's one thing I want to know is what is affected, but what isn't affected. And the other thing is it reversible?
Right.
And that would take a longer term study where you have the rats eating that diet and then you have to get them back on a whole foods diet and see if you can get recovery. And I do have a pilot study actually that it's unpublished at this point. It was just a small pilot study with 8 rats and they'd been on this junk food diet, this processed food diet for 8 or 9 months, so a long time, especially for a rat that lives only about 2 years natural lifespan. So after about 8 or 9 months, we took 4 of them and put them back on a whole food diet and the other 4 stayed on their refined diet. And in the first week or so, there were no differences between the 2 rats on the motivation test. They were both showing really poor motivation. But after 5 weeks back on the control healthier diet, those rats that were now on the control healthier diet actually saw improvements in their motivation when we put them back on the motivation test. So it looked like we got some rescue of motivation with a long enough time on a healthy diet. So that would be important to know. Can we reverse these things? Because if humans are starting to experience this eating for years like this, we want to know is, can we rescue that with just a lifestyle intervention, or would it require something even more dramatic, like pharmacology or something?
Smart Drug Smarts.
So thank you so very much to Dr. Aaron Blaisdell for taking the time for that conversation. Couple of things worth coming back to there. I didn't use the term Bayesian inference during the part of the conversation where we were talking about the types of deductions that rats might be able to make about the world based on incomplete information that they're given about the situation that they're in. But it sounds a heck of a lot for those of you that are familiar with Bayesian reasoning and probabilistic thinking, things like that, that's kind of obviously at a very intuitive level what animals are doing, how that processing might be happening sort of behind the scenes. Bayes was a mathematician from, I think, about 300 years ago, spelled B-A-Y-E-S. And I'll also drop a book recommendation that's almost like the book version of some of the topics that we talked about there, called Are We Smart Enough to Know How Smart Animals Are? It's a relatively new book, I think just came out in the last year or two, by Frans de Waal. I actually read that in the time since doing this interview. Kind of wish I'd read it first, but what can you do? Last but not least, there was his reference to that study done called the mark test in the 1970s, putting the mark of makeup or nail polish, whatever it was, on the monkey's forehead. That was done by Dr. Gordon Gallup. I hadn't realized that actually at the time that we interviewed Dr. Gordon Gallup on episode number 93 of this show. But he was the doctor who I interviewed in the great Smart Drug Smarts semen episode, which is, I know, a fan favorite for some of you. I did not put 2 and 2 together until after this interview had taken place, but kind of cool when these web of Smart Drug Smarts interviewees start to interconnect. But moving right along, let's continue with the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
So as promised, this is sort of a weird hodgepodge of interesting animal brain facts. The kind of things that you come across and you're not quite sure what to do with, but at some point they just need to come out in a podcast episode. And this is the episode, so here you go. So you heard during the past conversation how humans, despite having great brains, we don't have the biggest brains in the animal kingdom. So obviously, while size matters, size is not the be-all, end-all when it comes to brain complexity. And yet it does matter. And a great example of that is the spider. So most all spiders make webs. Webs are a pretty elaborate thing for an animal to have to do. It's not an easy thing cognitively speaking. And even though the size of spiders varies dramatically from tarantula-sized biggies to things that are barely small enough to see, the size of the spiders and the size of their webs kind of scales up and down, but the complexity of web building is about the same regardless of the spider's size. What this means is there's only a certain amount that you can actually crunch down the size of the spider's brain and still have it be able to perform web making. So there are some species of spiders that have gotten so small physically that their central nervous system fills up almost 80% of their total body cavity size, basically everything inside the exoskeleton, including in some cases about 25% of their legs. So little spiders' brains overflowing into their legs just so they can make their webs. Another animal oddball brain is literally interwoven with the digestive tract. There's a creature known as the colossal squid. This is the largest squid known on planet Earth. And the squid has a donut-shaped brain, including a hole in the middle. And when the squid eats, its food literally goes through that hole. The esophagus runs right down the center of the brain. The brain is not very big. It's less than a quarter of a pound in these squids. And apparently the squids eat very carefully, chewing their food into tiny, tiny, tiny pieces before they swallow, so they don't have any big chunks that injure the brain as they pass through this donut hole. We talked in that past interview about how octopuses have somewhat independently functioning limbs. A leech is another great example of an animal that's somewhat run by committee. The internal structure of a leech's body is divided into 32 segments, and each segment actually has its own brain. The neuronal ganglia from one brain are connected to the brain in the next segment, but each of these mini brains controls the segment that it lives inside, and they generally work independently of each other. So anatomists seem to agree that yes, this is a little animal with 32 brains. And finally, although there's often a lot of sexual dimorphism among species, where the males and females of a given species have big anatomical differences, typically these differences don't apply to the brains quite as much as to the rest of the body. Brains are generally around the same size between males and females, the biggest disparity being seen in something called the three-spined stickleback, which is a fish that lives in an Icelandic lake. In this species, males have brains about 23% heavier than their female counterparts. It's believed that this is because the males have basically sort of more to do in their job within the species. The male fish put on elaborate courtship displays, and they make nests. But before you guys in the audience start snickering and say, this species has bigger brains than the males, it's only fair to point out that in this species, it's the males and not the females that are exclusively tasked with raising children.
We like to think of ourselves as a digital speakeasy for brain hackers, but you can call us Smart Drug Smarts.
Okay, so that is the very end of episode number 170. Thank you for hanging around with us. If you're curious about the links to anything that we talked about here, that's online at smartdrugsmarts.com/170. And last week in episode number 169, I talked with Daniel Schmachtenberger about the future of medicine, big data technology, personal health, and of course, what all these converging trends mean for our brains. Next week in episode number 171, I think that we will probably be talking about flow, getting into flow, maintaining flow states, and the overall usefulness of altered states of consciousness. I spoke recently with authors Stephen Kotler and Jamie Wheel, both of whom are absolutely every bit as interesting to talk to as they are to read. That interview will be coming at you very soon, probably next week. We've got a St. Patrick's Day episode coming up for you in just a couple of weeks. A lot of good stuff on the near horizon, so stay tuned. I will be back with you next Friday. Have a great week in the meantime, and until then, stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.